While we ultimately want to build whole parts of the heart, the more immediate goal is reachable not in 5 to 10 years, but maybe in the more near future, is to be able to regenerate new muscle in an existing heart, in existing parts of the heart. And this could be tremendously advantageous in numerous forms of both pediatric and adult forms of heart failure, one of which, as shown here, is caused by heart attacks, largely in adults, where a coronary vessel is occluded, the blood flow to that part of the heart is lost, and those muscle cells die. And the result is the scar that you see on your right here that's indicated in blue, where the wall of the ventricle becomes very thin, and this is really fibrotic tissue. The heart has little or no capacity to regenerate itself, and so one is left with this scar permanently, and over time the heart deteriorates and gets worse and worse. And this is what 5 million people in America suffer from. Ultimately, the only course right now is a heart transplant for any of these types of heart failures at the end stage, but only about 2,000 heart transplants are done in the United States every year because of a clear shortage of donors. Now, what we know is that the heart is actually made up not just of muscle. In fact, only less than half of the cells in the heart are muscle. The other half of the cells in the heart are what we call fibroblasts, which are really just support cells for the architecture of the heart and secrete important signals to the neighboring muscle. But it's also those fibroblasts that get activated after injury and migrate to the site of damage and are the ones that lay down this scar tissue. And so we asked a few years ago, began to ask, if the heart has this great reservoir of cells already there in the organ, then might we be able to not reprogram fibroblasts to stem cells as Shinya Yamanaka did, but rather directly convert those fibroblast cells that are already in the organ into newly born cardiomyocytes. If we could do that, then we could harness this vast potential of cells already in the organ and turn them into new muscle cells for regenerative medicine without having to put new cells into the organ. And I'm sure you've heard and seen through so many grants all the hurdles that we are trying to overcome with cell-based therapies of introducing cells. I think we can overcome those with time, but there are clearly many hurdles we face there. And this might be a little bit easier. And so the approach we took was to go back to how nature normally builds a heart, which we had invested the last 15 years in trying to figure out those networks. And we asked if we essentially leverage that information and through the kitchen sink, if you will, of those networks at a fibroblast cell, could we get a cell to become more like a cardiomyocyte in a dish? And in fact, we were able to. And it turns out that a combination of just 3 key master regulatory genes, some of which I told you about earlier, are enough to turn a cardiac fibroblast into a muscle-like cell. And those were GATA4, which I mentioned causes human disease, TBX5, which causes a similar disease, and a third factor, MEF2C. Now, in a dish, these 3 factors could push cells to a cardiomyocyte-like state, but most cells were partially reprogrammed, and a few went all the way to become beating heart cells, even on plastic. But the question we really wanted to know is not could we do this on plastic, but could we do this in a living organism, in an organ, specifically in the heart in an animal. And so we did that experiment more recently. We tied off a coronary artery in a mouse, to mimic a heart attack. We injected the 3 factors with the virus, sort of a gene therapy approach, waited 4 weeks, and then took those hearts out and dispersed the cells from the heart into a dish so we could examine each individual cell and see how well we were able to reprogram the cells into new muscle cells. And about 50% of the cells look something like this. We genetically traced those cells that would start off as a fibroblast and become a muscle cell with a red marker that you see here. And about 50% of the cells look like this, where they looked essentially just like a real cardiac muscle cell, which is shown here. So that looked pretty good. And if we stain those with markers of the of the what we call sarcomeres, or the beating units in the muscle, it looks something like this. The green cell here is a real, if you will, or native cardiac muscle cell, and the yellow cell is a newly born cardiac muscle cell that used to be a fibroblast cell. And you can see that these also have these beautiful striations indicative of a cardiac muscle cell. But the real proof in the pudding in this situation is, can these cells beat? Because for heart cells, we have a very rigorous test of whether they're fully reprogrammed or not. And so it turns out that when we reprogram cells in the in vivo native environment, with all the right signals around them and the stretching, the mechanical forces they might experience, over 50% of the cells that are reprogramming go all the way to beating. And that's what you see here. If I play this movie for you here, this cell that's fluorescing green is genetically labeled to indicate that it used to be a fibroblast, or a non-muscle cell, and this is a real muscle cell that was already in the heart that we've taken out. And so now you can see when we electrically stimulate, both of these cells are able to contract, and as I mentioned, about 50% of the fluorescing green cells could be fully reprogrammed like this. And so we were very encouraged by this and proceeded to see if they could make a difference in vivo. But before that, we had to confirm one thing that's very important. It's not enough to make a cell, a muscle cell that beats in the heart, if it doesn't electrically couple with its neighbors. It would beat by itself, would not generate force, but moreover, be a potential source for rhythm disturbances. And so this experiment that I'll show you here is critical. And that is to determine if these cells can actually couple with one another, both the newly born ones, but also the previously existing one. So here what you see is a cluster of cells that we've isolated now from a reprogrammed heart in vivo. And now it's not a single cell, but there are 3 cells here, 1, 2, and 3. Only the red cell is newly born, and these 2 are old cells. And when I play this movie down here, we were looking at the wave of calcium that goes through these cells. And what you'll see is that when we stimulate, all 3 cells have the wave in synchrony, which means that they're electrically coupled with one another, and therefore they beat at the same time. And so with this knowledge in hand, then we proceeded to do the trial. Okay, but I forgot I was going to show you this movie. So just to illustrate for you what we've done here in a diagrammatic form, many of you, I suppose, have seen this Waddington diagram of this hill of a ball rolling down and a pluripotent cell becoming a specific cell type. And you're probably familiar with Shinya Yamanaka's work, where he took these balls, essentially, if you will, and rolled them up the hill back to a pluripotent state where they could go back into any one of these states. What we've done here is something different, where we've essentially taken these cells and jumped them over the hill, if you will, from one cell type in the adult directly into another without having to go back up the hill and then come back down. So this is something we refer to as direct reprogramming, because it's going directly from one adult somatic cell type to another. Okay, so here's the experiment then, the preclinical experiment, if you will. We took a large cohort of mice, did these infarctions, and then injected the reprogramming factors, and then at serial time points did echocardiography on the mice to see, even with this crude injection into the muscle, could we make a difference in their function. And at the end did MRIs to look to see, because that's really the best test to look at cardiac function. And so I'm showing you here a representative MRI on your left, and then the quantification across tens of mice in each category. And the dashed line here indicates the cardiac function by MRI of a normal mouse that hasn't had an infarct. This blue bar represents control mice that had an infarct but had a virus injected that was just a control virus encoding a control gene. And you can see that it's decreased. And then the green bar indicates the experimental mice that had the 3 reprogramming factors. It's clearly not normal, but it's much better than without treatment. And the MRIs, representative MRIs, are on the left. I won't go through right now. If we section these mice after sacrificing them, the controls look something like this, where you see now in a cross-section where the left ventricle is here, that this is the thin left ventricle wall with the scar tissue. And the treated mice look something like this, where there's still a thinning of the left ventricle, there's scar, but you can begin to see muscle interspersed now through this scarred area. And we could genetically label those cells and ask if those red cells that you see in this scarred area, thinned area, were just leftover myocytes or newly born ones. And we have genetically labeled these, and I'll just tell you that these, in fact, are all newly born and have the markers that tell us that these used to be non-myocytes, so they're actually newly reprogrammed cells. So we're very encouraged by this because we think that this represents a way to potentially regenerate heart muscle without putting cells in and all the associated hurdles, but rather harnessing the cells that are already in the organ and tricking them to do something that they would not normally do. Now, there's a lot of work to be done going forward to really understand this biology. And so with the help of a CIRM basic biology grant, we are now aggressively trying to understand the mechanism by which this occurs, meaning where do these 3 transcription factors that are controlling thousands of other genes, where do they sit on the DNA and what are they turning on and off that actually makes this happen? And what I'll just tell you from our early results is that it happens within hours. It's phenomenal how quickly the genome is being reprogrammed by these factors. And within 3 days, most of the transcriptional changes across the genome have already occurred. And then over the next several weeks, they're fine-tuning of gene changes that ultimately result in the cardiomyocyte phenotype. But the bulk of the activity is very, very early. We did not anticipate or expect that to be the case. And we have the tools now to actually understand the epigenetics and the transcriptional changes that occur at hourly intervals, and we're doing exactly that with your help. Now, even while we push to understand this and refine the technology, we're aggressively pushing forward with preclinical studies in larger animals, specifically the pig, to begin to see if this strategy could work in a larger heart that's more similar both in size and physiology to the humans. And again, this is with the help of the CIRM Early Translational Award that we're now able to do our studies in pigs to test both the efficacy of this approach as well as the safety. And so we've already initiated our first sets of pigs to begin to test how the delivery is occurring and what levels we're getting. And I'm very hopeful that in the next year to year and a half, we'll begin to have the results to see if this approach really can work in a larger heart. And we're very, very excited about this and are grateful again. This is not the kind of work we could do any other way. It's expensive, and NIH just doesn't support this sort of thing that's no longer in the discovery phase, but really is translational to move this into a preclinical state. Now, I'll just close by saying that this approach we're very excited about because we don't think it's unique to the heart. There's evidence at least in a dish now for many other cell types that one can directly reprogram an adult somatic cell into not just heart cells, but brain cells by folks at Gladstone, as well as Marius Wernig at Stanford, to blood cells, to liver cells, pancreatic cells. And it's our hope that others will now do these types of studies in vivo. And just last week, a group reported that they were able to convert in vivo cells in the brain, neurons, into spinal motor neuron-like cells that are lost in ALS. And so I think you can think of this strategy for multiple different regenerative approaches because the developmental biology of each of these cell types is now fairly well understood. And it's that developmental biology that then allows us to flip the switches, if you will, and tell a cell to become what we want it to become. And so I'll close there by just saying that I think the combination of powers of human genetics, along with the stem cell biology, particularly using reprogramming technology, puts us in a situation where we have unprecedented opportunity to both understand human disease in ways we couldn't before through disease modeling and leverage our knowledge to begin to think of new ways to regenerate organs going forward. And so I just want to thank members of my laboratory that are shown here that are just a brilliant group of people. Many CIRM scholars are in this picture that have been supported by our training grant at Gladstone, and all of them utilize our shared facility that you all have generously supported at Gladstone. And we've had great collaborators, including Shinya and his lab at Gladstone, Benoit Bruneau, who I mentioned, Bruce Conklin, and Sheng Ding, who moved to join us last year from the Scripps Institute. Thank you very much.